Well, thank you very much. I'd like to thank David Landsman and David Lippman and everyone else involved in organizing the symposium for inviting me to participate. I felt a little bit horrified at seeing the order of speakers and wondering whether I'd have any factoids left at all to share with you about the early days of getting the sequence databases, DNA sequence databases off the ground. But I'll sort of skim through and look for what might be different or novel relative to the others. I won't translate the title of my talk. I also quickly realized that you would have seen the same title 4 or 5 times in a row, so I'll explain where I got this in a slide or two. So we've been hearing a lot about the collaboration and U.S., Japan, U.S., Europe, and since I'm going to be nationalistic about my newfound home in Canada over the past few years, and just make the simple point that around the year 2000, Canada and Canadian scientists got quite concerned about the race to do big science in the context of genomics and proteomics, and set up something called Genome Canada and a number of affiliated genome centers, of which I'm affiliated with one, Ontario Genomics Institute, and have pumped about $1.5 billion into genomics and proteomics projects over the last 8 years. So I thought I'd try to find something as a new starting point, and so and I'll come back to this towards the end of the talk. In 1731, about 251 years before Genbank got started, something called the Library Company got started by Ben Franklin and a group of 49 or 50 friends, at which point he had drawn up instruments of association, and it was the first lending library in North America. It so happened it was private, but it was the model for public lending libraries that he went on to establish. And I took the title of my talk from the Latin motto that they have inscribed on their seal. This company still exists today, and translates briefly as to pour forth benefits for the common good is divine, which I think is a good precedent and model, and really resonates with all of the themes you heard in Bruno's talk and the other talks so far. So I'm not going to really dwell on all this, but just sort of setting a little bit of timeline. I believe the first nucleic acid sequence was published around 1965. It was an alanine tRNA put together with a lot of hard biochemistry and stitchery. Again, small on today's standards, but to show the sense in which the world was racing along. The first complete genome, so-called complete genome, was a phage sequence 5X174 in 1977, and that was a bit over 5,000 bases. And of course, there were many other sequences coming out at that point in time, because of the new technologies that Sanger and Maxim and Gilbert had established. And it really gave rise to a growing interest and enthusiasm and concern in the community about being able to get all these sequence data on databases. So I won't talk about the Rockefeller Workshop. It's been mentioned several times. I will note that there was a group at Los Alamos that was quite interested not only in the sequence data but also in the analysis. This included Stan Ulam, Bill Byers, Temple Smith, Mike Waterman, Walter Goad, and George Bell. And Michael and Temple both attended the Rockefeller Workshop, as did Howard Bolofsky, who, of course, was part of Genbank later on from BBN. So sort of passing into the more intense period of sorting out what should happen next, a really key point for the effort at Los Alamos was the arrival of Minoru Kanehisa from the East Coast of the U.S., who began working with Walter's group, and just everything A to Z, from entering sequences to developing software tools for organizing the data and sorting out how it should be organized and what processes would be involved. And I noticed a letter just recently that Walter had sent to Minoru about 10 years ago, saying that in looking back to those early days that he thought Minoru had as much as anyone to do with the start of the Los Alamos Sequence Library. So really tremendous impact on getting the effort going there. Shortly after the Rockefeller Workshop, the folks at Los Alamos were quite excited about the idea of building such a resource, and began collecting data and organized a small workshop where they invited scientists, I think for the most part from the US, to visit in around August of '79 to discuss the possibility of building up an analysis and database resource. This resulted in, or was meant to feed into, an unsolicited proposal that Los Alamos submitted to the NIH towards the end of 1979, where they proposed to build such a resource. That was ahead of the curve in terms of NIH deciding how it was going to do what it was going to do. There was also a lot of back and forth in the community and at the NIH about whether to fund and what resources would be involved in funding an analysis group as well as a database group. But I think there was a nice note of this from Bruno that they reached their 100 KB point in mid-1980 or late 1980 and had a celebratory party. One of the ongoing challenges in working the databases was trying to figure out how far apart and how many zeros to add on in order for the celebrations not to be so fast upon themselves that they were no longer exciting. There was a workshop, you heard earlier, that Europe was off and running. That and the fact that the community in North America was still quite concerned that something get going led to a workshop in mid-1980 in July, organized by Elke Jordan and Marvin Kassman, chaired by John Abelson. And came across a letter that Larry Kiedis wrote to Walter Godin and perhaps to all participants in the workshop on the second and last day of the workshop that sort of underlined their sense that they wanted to stop having meetings and get on with establishing the database, where he says the meeting adjourned with the optimistic expectation that there would never have to be another one. At sort of the end of the summer of 1980, Jim Fickett joined the group at Los Alamos. He was actually on the faculty as a mathematician in Texas, but got interested in what was going on in T10 and gave up his faculty job to come back and take on a postdoctoral role as an entree into the T10 group. And he very quickly settled into and got involved in a lot of the software tools that were involved in building up the entry and analysis of the data that was going on in the database then. So I think sort of ending this period of pre-Genbank was the formal publication as a lab technical report in early '82, March '82, of the Los Alamos Sequence Library database report that Jim, Walter, and Minoru put out, and that contained just under 500,000 bases at the time. And I just thought I'd sort of give a quick survey of some of the characters I've been talking about at this stage. Upper left-hand corner is George Bell, who with Walter Goat established the theoretical biology group early on, and Charles DeLisi was part of that at the beginning also. In the right-hand side, you see Temple Smith and Mike Waterman, who were at various times affiliated with Los Alamos and involved in that Rockefeller meeting, as I mentioned, and very interested in the sequence alignments, which of course the publications they put out later attest to. I believe this picture was actually snapped by David Lippman on a trip from Arizona heading back towards the East Coast in Los Alamos. And then the lower left, you see Minoru Kanehisa and Jim Fickett, who I both mentioned earlier. I think that's Jim in situ at Genbank and Minoru some years later. So getting into the phase one of the contract, from just the very early January of '82, I joined the T10 group, and I think my entree into the group was that they, though as best I could tell as I arrived there, were all brighter than me. None of them were card-carrying biologists, and they were eager in lining themselves up for the proposals they would be writing in response to the NIH RFP to have biologists on hand. So that made for a postdoctoral opportunity for me, which I was delighted to take and greatly enjoyed. So joined Walter's group and Jim and Minoru at that point in time. About a month later, the proposal was sent off from Los Alamos, and there was a quirk that we got caught up in, and I don't think this had been realized too far ahead of time, but it played out nicely in the end, or at least we worked to make it play out well, which is that since the NIGMS decided to do Genbank as a contract, Department of Energy, in its wisdom, thought it would be inappropriate for one government agency to contract with another government agency. And so we were encouraged to find collaborators who could actually, in working with us, be the primary contractors for the NIGMS. And so Los Alamos participated in two competing proposals, one through IntelliGenetics and the other through BBN Labs. And of course, as you know from earlier comment, Margaret Dayhoff and the folks at Georgetown University also submitted a proposal. So you've heard a bit about the deliberation process. The contract started in mid-1982. The proposal with BBN Labs was picked as the successful one, and that effort early on was led by Howard Belofsky, with Wayne Rendon and Fran Lewiter still leading the charge. Fran is now at the Whitehead Institute in charge of some of the genome informatics effort going on there. Walter Goad and Greg Ham had been, I think, in touch and intending to and beginning to collaborate from well before the contract was awarded. In fact, as was indicated, Greg was involved in helping to write the RFP on which the contract was based. So there was a close connection there. But shortly after the contract was awarded, Walter had organized a workshop at the Aspen Center for Physics, which involved a lot of the people who were interested in the sequence databases and analysis. And from Aspen, Walter and Greg wrote a letter to 5 or 6 different academicians in Japan, asking them and inviting them to participate in a collaboration with EMBL and Genbank, and asking if there are ways in which they could be supportive of or assist in making that happen. It actually took a while, as you heard a few moments ago, for that effort in Japan to take off, but once it did, it was a strong and third member of that collaboration. So just seeing milestones go by, 5 million databases towards the end of '84. A really significant aspect of the effort and a sort of play on whether or not the databases had time to really curate and annotate the sequences they were bringing in was the spin-off of initially an HIV sequence and analysis database that Jerry Myers undertook. He had spent a sabbatical at Los Alamos to work in and around the Genbank effort, but got very interested in HIV sequences, and he told me in an email a couple of weeks ago that his first jump into that was actually prompted by his daughter writing a report in high school and bugging him about pulling some sequences out of Genbank that she could include to make her report more interesting. And he didn't turn back. He stuck with the HIV sequences and went forward, and eventually secured his own funding and separately from the NIH to really do a very careful collection and annotation and characterization of all the HIV sequences. That's actually gone forward. Jerry's retired now, but Betty Korber, Kathy Mackin, and several other co-workers have carried that general trend forward now for a number of other sequences, including STDs, oral pathogens, human papillomavirus, and flu. So that ended up being a very strong branch off of the initial effort. So I think I thought I'd just close this section by mentioning that we became aware towards the end of '86 that DDBJ was getting off the ground, and came across a letter recently that I had sent to the folks at DDBJ, Takeo Maruyama, I believe, basically repeating the proposition that Walter and Greg had offered several years earlier, and with a lot of details about how they could begin interacting with us and the folks at BBN Labs also. So early '87, which was the end of the first Genbank contract led out by NIGMS, we're up to about 10 million bases. There was another competition again, and there's a key point here which I think in part addresses some of the questions that came up sort of along the lines of there were all these great ideas early on about what it would take and how one should keep up with the data. And the project really was resource bound in the first few years. We had put in a proposal that was a match to the amount of money that we understood to be available, not a match to what we thought it would take to get the job done. We worked within those bounds for the first few years. We had a number of stormy meetings with our advisory board where they expressed their concern about our not keeping up with the literature. Don Lindbergh showed one quote from me remembering some of those moments. And I think the real turning point, which was a significant turning point as we went into the second contract period, was that Jim Cassatt, who was the new program manager from NIGMS, worked with us and with the NIH system and eventually led to Jim Wengarden putting in a really significant up-ramp, I think in the vicinity of $4 million a year to go towards supporting the Genbank contract. And that represented at least a five-fold increase over the resources that have been available for the first 5 years, and at the end of the day made a tremendous difference in our ability to catch up with the literature and with making the data available to the community. So in this go-round, we were in 3 competitive proposals, one with IntelliGenetics, one with BBN Labs, and a third with DNA Star, including Fred Blattner and Temple Smith in that go-round. In this case, IntelliGenetics was the successful proposal paired with Los Alamos. That was led by IG's CEO, Mike Kelly, with initially David Christopherson, but very quickly David Benton coming in as the program manager at IntelliGenetics. Significantly at this point for us, Paul Gilna and Tom Marr were recruited into T10. Tom was already at the lab in another group, and Paul Gilna at the University of Chicago. And Paul and Tom more or less took on the roles that I and Jim had supplied under Walter Gode's leadership. I was sort of the biology guy, Jim was sort of the computer guy, and that were the hats that Paul and Tom took in going forward. Paul Gilna played a very critical role in working with the journal editors, similar to what Graham Cameron was describing for both himself and Patricia Kahn at EMBL, and working with our advisory board to track down leads and any means of persuasion that would help with getting journals on board with direct submission of database stuff. Soon after that, a joint international advisory board was established overseeing all 3 databases. I'm skipping over a lot of work and a lot of flurry, but really significantly over this middle period of this contract was that we really did turn around the amount behind the literature we were and really switched over to the majority of the data coming in through direct and electronic submission rather than through manual data entry. Towards the end of that contract, I took on a role as group leader for the theoretical biology group and turn over the leadership of the project to Paul Gilna and Mike Sinkowski. Mike had been with the database all along, working with initially Jim Fickett and then Tom Marr, but took on the role of overseeing the computational work. And Mike really played a significant role in a lot of the changes we were going through in computer hardware, going from customized code dealing with text databases or files to working with the Sybase relational system during that time period. So towards the end of that period, we're up to 90 million bases, and of course, as you heard earlier, the database went over to NCBI at that point in time. I thought I'd just throw out a few more pictures here. The upper left-hand corner is a picture in the beginning of the first contract with Walter, myself, a guy named Randy Linder, who was a summer student who's now on the faculty at the University of Texas as a member of the Molecular Evolution Department. Lower left-hand corner is Howard Belosky, who led the effort at BBN. Lower right is Tom Marr, who was at Los Alamos as part of the second contract in particular, went on to Cold Spring Harbor, then to found a company called Genomics, then on to University of Alaska to take on a bioinformatics chair, and recently is back in Colorado with another startup under his belt. The upper right-hand corner is the Genbank team, and I won't name everyone, but this is the one picture I could grab easily that you can see Michael Sinkowski and Paul Gilna. Those who know them have found them already in there, I'm sure. And then finally, NIH actually had a wonderful symposium in '91 to honor Walter Goad and his effort in getting the Genbank database off the ground. And these are the database groups from IntelliGenetics and T10 in Los Alamos who, on the occasion, sent a message to Walter. Hello, Walter, as you can see here. And again, nice selection, though quite a subset of all the people that were involved in getting it off the ground. So I just thought I'd spend the last 2 or 3 slides on just some of the transitions that we went through. In addition to just the notion of keeping up with the data and all of the interesting biology associated with it, there was quite an upheaval in technology during the 10 years that we were involved with the project, or 10 plus years, including the sequence library. We went through big changes in server hardware. We went through big changes in operating systems, both on the hardware and operating system front. One of the ironic things at the end of the day was that in order to do what we had to do and do it well, we figured out that we should get off of the massive mainframe system at Los Alamos that had been part of the reason that one would want to be at Los Alamos in the first place, and move on to computers that we had more direct control and that were more comfortable for the aspects of editing and managing text files and the database systems associated with them than the heavy-duty number crunching codes that were more the reason to be on a big mainframe. And then finally, data management ended up moving over to relational database management system around 1990. It's interesting to note that one of the first debates between Minoru Kanehisa and Walter Goad, when Minoru arrived at Los Alamos, was Walter was aware that there was a test relational database management system up on the Livermore time-sharing system at Los Alamos called Framus. And for all the good reasons that would want to be in a relational database, Walter thought it would be great to put the sequence library into that system. Minoru, looking through it, thought that the system wasn't at all adequate for dealing with the kind of data that were going to be in the sequence database, and really pushed hard to stay out of Framus, though Minoru did go ahead to publish a paper about the experience of and appropriateness of relational database management systems around that time for sequence data. So it took the practicality of relational systems about 6 years to catch up with the concept, which was very appealing. Community interface also went through a lot of transitions. Again, not going to go read through all of these. But the way we distributed the data went through a lot of different cycles. Some of these rose and fall in short order. Some rose and stayed on for quite some time as it made sense. And the same point is to be made about data collection, how we got the data, starting with manual entry, which was talked about earlier. And I think the annotations that one saw on instructing data entry folks on how to read through a sequence really is to the point. Quite complicated at times, but also quite time-consuming. And as Graham pointed out, the fact that by this point in time, most of these sequences were starting in computers. We switched as rapidly as possible, and with the help and the leadership of people like Rich Roberts in particular in encouraging us, but also encouraging the journal editors of the world, including the journal he was affiliated with, to move in this direction. So these are quite significant transitions, and I think at the end of the day, in spite of all the technical things, I think what I would think of as the social change or the cultural change in science of separating the DNA sequences from being in the same direct pathway of journal publication with this pseudo-parallel pathway of being submitted in advance of publication in an electronic form to the databases was very significant. And I think in terms of a paradigm shift was maybe the most significant for us. So drinking from the fire hose, which is how we felt on most days in terms of the rate at which sequence data were coming out. There were a number of paradigm shifts. I just mentioned the electronic data publication as one. Database quality assurance, this was hotly debated on our advisory board. It was hotly debated by database staff, and it was certainly hotly debated in the community. Early on, we had a very strong editorial role, and we shifted as we had to, and after getting the go-ahead and the resolution from our advisory boards to really focus on just making sure the sequence data we're getting in and we were keeping up the literature and sacrificing the editorial and curatorial components of going over the data and making sure that one data entry was consistent with the other and that they all could be fit together in a nice way. And then finally, data integration revisions and updates. That's shifted quite significantly over the last few years. Certainly in the last few years of the database from, again, being a role where we tried to impose order on the entries and perhaps in the spirit of Rich Roberts' suggestion, really end up shifting more to an archival role where the community was free to and encouraged to let us know when something needed to be changed or combined or fixed or corrected, but we didn't go looking for those things ourselves except for when they could be detected by automated means. So I guess the last couple of slides, one at various times, I thought it'd be interesting to go back and look when and if we ever made predictions about how much data there would be. The first two instances, we were pretty close within the confines that we had. Jim Fickett and Minoru and Walter made the statement that there could be as many as 10 to the 7th bases within 3 years, and they were quite close to that mark. Jim and I made a statement in a later publication about there being close to 10 to the 10th bases in 10 to 20 years, and again, ended up being pretty close to the mark. Both of those were offered, and I think based no more than quick conversational sort of, there could be as many as thoughts. In the third example here, with the benefit by then of early discussions about genome sequences and the possibility of doing the genome, I actually went through and tried to build in real estimates based on real notions of how much data would have to be collected in order to do the genome in the time frame that was being suggested. So the somewhat realistic modeling ended up being the furthest off the mark at the point in time when that was made. So I thought just as another sort of numbers game, I'd say not so much making a prediction how much sequencing we'll be doing, but beginning to wonder what are the upper bounds or how much will be enough in some directions. And this isn't meant to be exhaustive, but including the WGS section, we're up at about 2 times 10 to the 11th bases in one of the recent releases. I thought it'd be interesting to at least conceptually ask how much DNA, how many genomes are there on the planet and how many bases in those genomes. If one said you were going to sequence all the DNA, and I'm ignoring generations in time here and just taking a current snapshot of the world, if you backed off from that picture, which I don't supply a number for because there are a fair number of unknowns there, but if you said in a more limiting way you wanted at least one genome sequence for all described species, there's still a fair amount of back-of-the-envelope guesstimation there, but one comes up with a number a little over 3 times 10 to the 15th bases. If you said, well, let's not limit ourselves to described species, but make a guess at how many species there are that haven't been described yet, one goes up about an order of magnitude in the number of bases that would be included in that. If you then, on top of that, said, well, what we're really interested in population variation within a species, then you go up another 2 orders of magnitude to 3 times 10 to the 18th. A project that we're funding now and been quite interested in is DNA barcoding. And that takes a different stance for those of you who are familiar with it, sequencing 650 bases from the mitochondrial genome of any eukaryotic species in order to be able to uniquely determine the species in the future by comparison to the reference database. Their general approach is based on doing 10 specimens per species. So if you said how much sequencing would it take to barcode every species or all of the described species or all of the estimated species, you end up in the 10 to the 11th or 10 to the 10th domain, which is actually on a par with how much data there are today. So not too strange to estimate. So my last slide here, Francis, is going back to the data, the library company that I mentioned at the beginning. When they got started, actually about, I guess, 60 years after they got started. They built a building to house themselves at this location in the Independence District of Philadelphia. You can't see it in the detail in this painting, but there's a nice statue of Ben Franklin recognizing his role in getting it off the ground at the top there. At some point after that, the library company needed to move off, about 100 years later, move off to other headquarters, but another institution that Ben Franklin founded called the American Philosophical Society, which you saw Bruno cite earlier on today, decided to acquire the property and built a facsimile building on the same site in which to house their library for the American Philosophical Society. And it's a nice point of residence that Walter Gode's papers are now resident in that building. And anyone here with due notice and willingness to observe all of the rules that they associate with getting access to the papers can go and sift through all of Walter's papers and records in getting the Los Alamos Sequence Library and Genbank off the ground originally. So I'll close there, but just as everyone else would acknowledge everyone who's helped out on every front in the past with DDBJ, EMBL, Genbank, and its several partners. I counted that I've moved, I think, offices 10 times and homes 6 times since I first started with Genbank. So though I was delighted to have this invitation, I was also panic-stricken because I'm down to maybe one file folder about a quarter-inch thick of any records whatsoever of the early days of Genbank. So I relied on sending emails and phone calls out to a number of people who were very generous in sharing information back. Brian Gregory at the University of Guelph helped with the genome size estimations, and finally the APS library staff and Lana were helpful. And I just want to say, both retrospectively but going forward, it has really been wonderful to see how Genbank has flowered under NCBI's stewardship over the last few years and the continuing collaboration with their partners. Thanks.